This video presents the standardized bowl trapping technique for day-flying bees and the terrestrial habitats. This was made as a supplement to the Handy Bee manual and should not be used as a sole resource. A list of resources is listed on screen, and links to all the resources can be found in the description below. The Handy Bee manual is updated frequently, so please ensure you have the latest version. For specialized habitats, please refer to the Handy Bee manual, U.S. National Native Bee Monitoring Research Coordination Network publications, and other literature. Colored bowl traps, or bee bowls, are apt tools for the inventory of bees as they are inexpensive, easily obtained, require no entomological training, and may catch small bee species not easily captured by net. Soapy water bowl traps are small fluorescent blue, fluorescent yellow, and white colored plastic cups that are filled with water and liquid dishwashing soap. Studies in the eastern North America have shown that the size of the bowl does not matter. The current standard is to use a 3.25-ounce souffle cups, as they are large enough to be seen in the field and not get lost in the grass, but still small enough to be held in one hand. Bowls deployed in hot, low-humidity environments will lose water very quickly, so larger bowl sizes may be used instead. Please refer to the Handy Bee manual for instructions on purchasing and painting bowls. Insects are attracted to the bowl colors as they mimic flowers and land on the water surface. The dish soap decreases the surface tension of the water, thus causing the insect to sink beneath the surface. Most insects die within 60 seconds of being immersed in the soapy water, but can revive if removed too soon. Bee bowls should be placed in exposed settings. Avoid placing traps in heavily shaded areas, such as under trees or thick herbaceous vegetation, as few to no bees will be collected in those bowls. If the vegetation is very tall, consider using bowl stands to elevate the traps to vegetation height. Bowls should be clearly visible from above. Think bee's eye view. Bowl deployment and collection should ideally be conducted between 9 a.m. and 3 p.m., or while the sun is up, unless your specific project goals require sampling outside this time frame. Bowl traps must be active for a minimum of 6 hours, maximum of 24 hours. A longer trapping period is ideal, and it is important to maintain consistency in active trap collection times throughout the project. Record trap deployment and retrieval times, as these serve as an effort indicator in the data. If no bees were collected during the survey, it is important to document that data on your survey sheet. At least 2 of these conditions must be met throughout the sampling period to deploy bowl traps. Temperatures above 55 degrees Fahrenheit. Cloud cover does not exceed mostly cloudy, defined as more than 75% of the sky is covered by clouds. And winds are mild, or less than 10 knots or 3 on the Beaufort scale. Do not deploy bowl traps during or right after precipitation, or if precipitation will occur while the bowl traps are deployed. Before you begin, it is good practice to pre-fill survey sheets and tracking labels to reduce time spent writing in the field and ensure data consistency throughout transects and sites. Tracking labels are small pieces of paper that contain the minimum data required for tracking samples during specimen processing. They are placed in each Ziploc bag with specimens after each survey. Tracking labels need to be printed using a laser printer on heavy cardstock, or ideally right in the rain paper. Tracking labels should only be filled out using a pencil. Never use a pen or marker on the label or on the outside of the bag, as the ink will dissolve. It is also good to create a supply checklist to confirm you have all the necessary supplies. Refer to the Handy Bee manual for a list of recommended supplies and a tracking label template. Prepare at least one 1-gallon jug of soapy water per transect you will survey. Do this by filling the jug with water and mixing in 1 squeeze of unscented Dawn dishwashing liquid. In addition, sort your bowl traps into sets for each transect you will survey to save time in the field. Alternate between fluorescent blue, fluorescent yellow, and white bowls. Make sure you have 30 bowls for each transect, with 10 bowls of each of the 3 colors. Stand at the start of your established transect. Place a bowl down at the start of the transect. If the vegetation is tall and obscures the bowls, elevate the bowls to the height of the surrounding vegetation using stands so that the bees are more likely to see the bowls. Otherwise, set the bowl on the ground. If absolutely necessary, you can flatten vegetation just around the bowl to increase the bowl's visibility. Fill the bowl to about 3/4 full of the soapy water solution. If there are 2 or more surveyors, it is sufficient to have one person set bowls down while the other person fills the bowls with the solution. Continue setting and filling bowls 5 meters apart, alternating between fluorescent blue, fluorescent yellow, and white bowls so that no 2 bowls of the same color are next to each other. Continue until all 30 bowls are placed. The bowls do not need to be laid out in a strictly straight line. The transit can meander around tall vegetation or other obstacles so long as the traps remain 5 meters apart. You can use a measuring tape to measure 5 meters, or you can measure out 5 meters and count how many of your steps measure 5 meters. After you finish setting out all the bowls, ensure your survey sheets and tracking labels are filled out with all necessary data. Do not leave until you've recorded all your data. Upon arriving at your survey site to retrieve the bowls, record all necessary data on your survey sheet and finish filling out your tracking labels. Note any traps that have been disturbed, tipped over, dried out, destroyed, or otherwise rendered unusable. Also note bowls that collected no specimens. To start, go to the first bowl in the transect. Pick up the bowl and strain the contents out using an extremely fine strainer, brine shrimp mesh aquarium net, or net bag. Do this over another container in case of any spills. If there are still insects stuck in the bowl, rinse and re-pour the bowl until everything in the bowl has been collected. Continue collecting bowls and straining insects out until all the bowls have been collected. Unless your specific project is specifically studying the differences between bowl colors, pool the insect specimens across bowl colors. Using your fingers or a spoon, remove any large plant material from the bowl. You may also remove any large, obviously non-hymenopteran insects, such as butterflies, slugs, or snails, but be careful to check for any bees or other small insects that may be attached before removing the contaminant. Move the specimens from the strainer into a quart-sized Ziploc bag or whirl pack. Use your fingers or a spoon to scoop out any specimens that do not easily go into the bag. If you are surveying in a remote location and will be in the field for an extended period of time, you can add a few drops of alcohol to the bag to preserve the specimens while you continue surveying. Alternatively, you can transport the bagged specimens in a small cooler with ice. The choice of preservation method from field to lab will depend on your project's objectives and the preferences of the lab that will be processing and identifying the specimens. Gently roll up the bag, trying to remove as much air as possible without crushing the specimens, then place inside another quart-sized Ziploc bag, along with a fully filled out tracking label. The label can also be placed inside the specimen bag as long as it has been filled out using dark lead pencil. Before leaving, ensure all data has been recorded on all necessary sheets and you have all the supplies you brought with you. The specimens should be stored in a negative 20 degrees Celsius freezer as soon as possible to prevent the specimens from deteriorating. The specimens should be processed within 6 months of capture. As a reminder, this video presents the standardized bowl trapping technique for day-flying bees and their terrestrial habitats and is not intended to be used as a sole resource. For more information on bee collections and links to resources, please refer to the handy bee manual or any of the other resources below. We also have other videos on bee survey techniques which can be found on screen. Thank you for watching!